So, hello, good afternoon to everybody. Obviously, it's a bit of a challenge to give an overview on neurodegenerative disease, and I'll try to do that as compact as possible. And please excuse that I probably go pretty quickly through the slides, but if you have specific questions, we have an opportunity after the session to talk outside of this room. So when we talk about neurodegenerative diseases, it's really a broad spectrum that we have nowadays. Obviously, the prototype is Alzheimer's disease, the most frequent one, but then there are others as well. In the area of dementia, there's also frontotemporal dementia, which is a subtype of these frontotemporal lobar degenerations. Recently, it has been recognized that it is often occurring jointly with motor neuron disease, which is interesting because that illustrates that there may be a common underlying molecular mechanism in diseases affecting several parts of the nervous system. Then we have the diseases with Lewy bodies, most prominent amongst them Parkinson's disease, but also dementia with Lewy bodies. And then there are diseases that are depending on prions, where proteins are the templates for developing the diseases rather than genetic and other reasons. And then there is this group of polyglutamine repeat diseases, Huntington's chorea being the most prominent of that, which probably also share a common mechanism. Obviously, as said, I will only be able to address few examples, but I try to illustrate the principles there. When it comes to imaging techniques, we face a similar problem. It's a broad spectrum nowadays. Imaging started more or less with looking at structure and blood-brain barrier, and nowadays that's the domain of magnetic resonance imaging, including dynamic imaging, but also there are aspects of blood-brain barrier that are more complex, for instance, the PGP function, which can be addressed with PET. Then we have specific molecules, typically proteins and deposits of proteins that play a pathological role, and they can be addressed with PET, to image with PET, but there is also the nanoparticles, should be nano, sorry for that typo, nanoparticles or the liposomes. Then synaptic function, of course, that's kind of the downstream thing. We are very much interested in neuroscience because that's what the brain is all about. And we have quite a number of measures for that, which can be coupled to the more molecular aspects. And then, most recently, the interest has turned a lot into the connections, really, and I will also show a few examples. fMRI obviously plays a role there, DTI, tractography, and then, of course, the transmitters, which are responsible for the connections to be studied with PET and SPECT. So why do we do imaging? I think imaging plays an enormously important role because we all know the progress in genetics, it's a big progress there, but there is a huge gap between what genetics does and what genetics illustrates and how that ultimately affects what we are interested in the clinic, cognitive impairment, motor impairment, dementia, and so on. So there are multiple steps in between. And really to bridge that gap, that's where imaging really sits. And there are a number of things I'll try to illustrate in examples how we can address that. This is a very busy slide. I would just like to use it to illustrate another point, which again is very relevant for imaging, is that the brain obviously is not a homogeneous structure. It has many, many substructures, and these substructures interact with each other. And for instance, when we look at Alzheimer's disease, we know from basic science that there are certain brainstem regions like locus coeruleus, reticular formation, basal forebrain with the cholinergic nuclei, raffinucleus, serotonergic, which have diffuse projections into the cortex and affect particular areas in the cortex, regulate what the cortex does. And the fascinating thing is that with imaging, we actually can look at the interplay of these things. use transmitter markers to look at these aspects, and look at functional imaging to look at what's happening here on this side. So that's why multimodal imaging plays such an important role and what can be done with that. Few examples. Simple glucose metabolism. We nowadays know that it is linked to glutamate, glutamine cycling, and not only to energy metabolism. So it's one of our best indicators of synaptic function for that reason. When we look at Alzheimer's disease, we typically see atrophy on an MR scan, and we see low glucose metabolism in the hippocampus. We see low glucose metabolism in posterior cingulate, which should be that hot in a normal subject. And that's a frequent finding, but it's not always the same. Sometimes in patients we see that. It's even more severe disease, but the hippocampus looks pretty normal. But we now see glucose metabolic impairment more in the neocortex, and especially in this precuneus area, the posterior parts of the cortex. So we realized from the neuroimaging that there are different subtypes of Alzheimer's disease, and they especially appear at an early stage of the disease. And actually that illustrates that imaging can demonstrate and analyze what's happening, and will classify these subtypes and analyze what's happening there. And it's also, of course, relevant because what is typically seen in the transgenic animal models is most closely related to these very few autosomal genetic human forms of disease. Whereas the most frequent form of human disease is this amnesic form, which is typically has a late onset and has other cofactors like vascular cofactors and things like that. That was the first example I was showing you. Or we can also see typically with the early onset patients who start with more neocortical dysfunction. So the interesting thing is, why do they end up at the end when pathologists look at it in nearly the same domain? And it looks very similar to pathologists, whereas it starts very differently. Maybe the link is the connections between these areas. So there are certain connections between the hippocampus, entorhinal cortex, and the neocortical areas. And again, with imaging, we can study these areas by tractography, for instance, and can demonstrate that these tracts are affected. And again, the interesting thing is to really analyze how these different pathways of disease relate to each other, the role of the tracts play, and what's happening there. Obviously, there are a few key areas in the brain. Posterior cingulate is one of them, and entorhinal cortex would be another, that probably mediate what's happening in the neocortex to what's happening in the hippocampus. Talking about neocortex and hippocampus, of course, there's amyloid. Amyloid is currently the big clinical story in human Alzheimer's imaging, because we now have tracers for that can be used clinically. Pathologists have always pointed out that in most patients this follows a certain rule, mostly concentrated on these association areas. And interestingly enough, there's not much amyloid deposition in the hippocampus in humans, whereas there is a lot in the experimental animals, which is one of these reasons why we aren't convinced that the models really reflect human disease. I will come back to that. So the current hypothesis, the prevailing hypothesis is that the amyloid starts off and then the atrophy comes later, and that's actually true in most patients at least. And we have looked at that in a multi-center study and demonstrated that it can be done nowadays in multi-center environment, which would qualify that technique as a biomarker. Those who have heard the previous presentation, the previous session, know what it means to be qualified as a biomarker. It's easy relatively easy to define the normal range, because in normals, at least in not too old normals, it's a bimodal distribution, so there are few normals who have amyloid, and of course that's an interesting subsample, but it's easy to define where the majority of normals sit. And when we use that, and in those follow-up studies that are currently available, it is demonstrated that it actually has a high predictive value at the stage of clinical MCI, mild cognitive impairment, patients not yet demented, but already showing amyloid deposition, and those who are already showing amyloid deposition frequently proceed to develop dementia. Typically, on average, after 2 or 3 years in these samples, but that varies depending on the stage of the disease, whereas those that don't have amyloid deposition very rarely proceed to dementia, and especially not to dementia of the Alzheimer's type. In international series, it's not always 100%, it's typically around 90% negative predictive value. It's also interesting that the amount of amyloid, once it's pathological, once it's above normal, doesn't play a major role. So it's really more or less a yes or no story. If there's amyloid, it's bad, but how much we poorly understand so far, and the factors behind that we do not yet understand so far. It can also be used in intervention studies, and it has been used to study whether drugs that are supposed to remove amyloid actually do it, and it has been demonstrated that at least some of them do that. But the interesting question is, does that have downstream effects? Does it actually improve synaptic function if we do that? Again, that's clinical outcome, but not only clinical outcome. We could also use imaging methods of synaptic function to study that. There's another aspect to amyloid that is so far rarely been studied, is how the amyloid is being removed physiologically. And that probably has to do with transport mechanisms and blood flow. And I think it's another big area where imaging will show us in the future what's going on. There are certain proteins involved in this A-beta removal, and some of them are probably accessible also to imaging. There is the other story. It's not only amyloid, it's tau. And the interesting with tau is that tau really begins mostly in the hippocampus and associated areas, and then spreads to the cortex. So it's kind of complementary. So we have a story, at the end they come together, but they start in different brain areas. So again, what's the connection there? And we should study that. There are now efforts to image tau more specifically, and there are tracers being developed. I can't go into the details of that. A few of these tracers, and especially one of them, is available for clinical use and has been compared to the amyloid tracers with some interesting results which still need further clarification. Then there's the alpha-synuclein, which is now being addressed, the protein that is important for Parkinson's disease and Lewy body dementia. Again, new tracers coming out. And then with the Lewy body disease, the downstream effects, if that story is true, would be the dopaminergic system. And again, the dopaminergic system is actually a system that has been very well studied over the years, including our chairman. And that's something where we know a lot about the details of turnover, specific enzymes, and now also increasingly the transporter and their role transporting dopamine from the substantia nigra to the brain. So that's also an area that is already well developed and will increasingly be linked with the other imaging modalities. Cholinergic systems, of course, are of interest, especially in dementia, and that's illustrating the anatomy starting in the forebrain with diffuse projection in the cortex. When it comes to development of tracers, that has been a little bit of a frustrating area because there are a few that work clinically for the acetylcholinesterase, but that's only at the end of the process, whereas it has been more difficult to develop something at the production side. We still don't have anything for the choline acetyltransferase. We have traces for the receptors. Most interest is now concentrating rightly on the nicotinic receptors because they have been demonstrated to go away in dementia, and maybe we will see a few more coming up and clinical results coming up with these traces on the nicotininergic system. This would be quite important to understand what's happening in Alzheimer's disease, depending on as a result of the deposition of the pathological molecules. That's some work we have done with the acetylcholinesterase analogs, demonstrating that it mainly affects the cortex while the nuclei still survive. So it's, again, a synaptic and transport problem rather than something that becomes most prominent at the nuclei at the beginning. Another area of active research is the immune system, and in the brain it's the microglia that are the resident immune cells, and microglial activation happens in lots of brain diseases as a reaction to nearly any damage of the brain. But we need to understand what the microglia is actually doing, because it can increase neuronal damage as well as contribute to removal of pathological proteins. Imaging, typically again done with PET. There is a traditional tracer, the 11-CPK-11195, that's being available for clinical studies and has been demonstrated in specific diseases, for instance in multiple system atrophy, to show a local signal in those areas affected. In MCI, some patients show increased signal, others don't, as illustrated here. There is a link to systemic inflammation that we do not yet fully understand with inflammation in the brain. We know that systemic inflammation impairs the prognosis of patients with neurodegenerative disease, and we should better understand how the systemic inflammation does that. There are new tracers being developed, and they also surprisingly showed an interesting phenomenon, that there's a polymorphism in humans. Some of these tracers do not bind really because the receptors are in a particular state in those subjects due to that polymorphism. Which is interesting in its own right, whether this polymorphism is just a normal polymorphism or whether it's actually associated with some psychiatric diseases. There are hints to that. That's a recent development. Currently, it's more a confusion to the field, because it has to be taken into account when using these new tracers. And so we have to look at that polymorphism in order to interpret the results. Last example, something that's currently coming up, interest in imaging transcription and regulation of transcription, because transcription not only plays a big role in cancer, it also plays a big role, obviously, in neurodegenerative disease, and is often downregulated in neurodegenerative disease. So there are tracers being developed for measuring the activity of histones, and you will see also papers at this meeting about that. And that is an example from ongoing work mainly from MD Anderson, where it has been demonstrated that HDAC activity and its inhibition by an HDAC inhibitor, histone deacetylase inhibitor, actually can be imaged, and the activity of that inhibitor in the brain can be measured, which is so far has been a big challenge. So in summary, I tried to show you some examples, and obviously it was only examples, what imaging can do in the neurodegenerative area. And I would like to stress that actually a key issue is that it can do the longitudinal things, because all the ex vivo methods, which often are more accurate and can produce more molecules or study more molecules at the same time than imaging can, all these ex vivo methods cannot do the longitudinal studies. So doing longitudinal studies is one of the key things in experimental as well as in clinical applications of imaging. Then I showed you that imaging can contribute to differentiation of disease that is not obvious from a pathological point of view, but may be very important to really understand how the diseases develops, and Alzheimer's subtypes is an example of that. I think we will learn a lot more about the formation and the removal of amyloid by using these imaging techniques in the future, and we certainly have to have a closer look on the vascular factors than has been happening so far. And of course, there is a big interest in the shared mechanisms of neurodegenerative disease, mechanisms that are not only relevant for Alzheimer's, and there are plenty of shared mechanisms. Inflammation as a modulator, synaptic plasticity, we will have a presentation here on that. Brain resilience, which is kind of something related to brain plasticity, how the brain maintains its reserve, facing a challenge. We barely understand that so far. We know it exists, and we should use imaging to study that better. Tau deposition, new traces coming up. It's certainly a difficult, challenging area. We would like to be able to image axonal transport, and you will have some papers at this meeting about that. Oxidative and mitochondrial damage is always a big issue in neurodegenerative disease, and new traces and approaches are needed. And then, as mentioned, translation and transcription is an emerging topic. From an even bigger view, why do we do imaging? And as said, of course, we want to look at specific molecular changes in specific brain regions. But that's not all, because that also can be done by ex vivo methods, and it sometimes can be done more accurately. So what we can do with imaging really is to look, in addition, at the networks, at the interactions and the dynamic interactions between these brain regions, and to, at the same time, study the molecular changes and the functional and structural downstream effects. So that's why multimodal and multidimensional imaging is so important for understanding pathophysiology. And ultimately, of course, it's all about improving therapy. And what is lacking so far in many studies is really to do a targeted intervention and then check whether that has not only the targeted effect, but also how it affects other systems. So that would really be practical systems biology. And that's what we really should be doing, using our longitudinal dynamic systems, imaging systems, to study systematic interventions and their effect on these systems. And I think that's the big perspective really in neurodegenerative disease and imaging. Thanks for your attention, and as I said, happy to take questions afterwards.